Now let's talk about the peripheral tolerance. Several mechanisms silence potentially autoreactive T and B cells in peripheral tissues. These are best defined for T cells. These mechanisms include the following. Say several mechanisms silence potentially autoreactive T and B cells in the peripheral tissues. These are best defined for the T cells. These mechanisms include the following. 1. Energy. Lymphocytes that recognize self-antigens may be rendered functionally unresponsive, a phenomenon called energy. Energy. We discussed earlier that activation of antigen-specific T cells requires two signals. 1. Recognition of peptide antigen in association with self-MHC molecules on the surface of antigen presenting cells and a set of co-stimulatory signals called second signals from the antigen presenting cells. The second signals are provided by certain T cell associated molecules such as CD28 that bind to their ligands. The co-stimulators B71 and B72 on antigen presenting cells. If the antigen is presented to T cells without adequate levels of co-stimulators, the cells become energetic. If the antigen is presented to T cells without adequate levels of co-stimulators, the cells becomes anergic. That means they become unresponsive because of co-stimulatory molecules are not expressed or are weakly expressed on resting dendritic cells in normal tissues. The encounter between autoreactive T cells and their specific self-antigens displayed by these dendritic cells may lead to anergy. Several mechanisms of T cell anergy have been demonstrated in various experimental systems. Several mechanisms of T cell anergy have been demonstrated in various experimental systems. One of these, which has clinical implications, is that T cells that recognize self-antigens receive an inhibitory signal from receptors for that antigen that are structurally homologous to CD28 but serve the opposite functions. The T cells that recognize self-antigens receive an inhibitory signal from receptors that are structurally homologous to CD28 but serve the opposite functions. Two of these inhibitory receptors are CTLA4, which like CD28 binds to B7 molecules, and PD1, which binds to two ligands that are expressed on a wide variety of cells. Because CTLA4 has higher affinity for B7 molecules than does CD28, CTLA4 may be preferentially engaged when the levels of B7 are low, as when antigen presenting cells are presenting self-antigens. Conversely, microbial products elicit innate immune reactions during which B7 levels on APCs increase and the low affinity receptor CD28 is engaged more. Thus, Affinities of the activating and inhibitory receptors and the levels of expression of B7 may determine the outcome of T cell antigen recognition. The importance of these inhibitory mechanisms has been established by finding the importance of these inhibitory mechanisms has been established by finding that mice in which gene encoding CTLA4 or PD1 is knocked out develop autoimmune develop autoimmune diseases. Furthermore, polymorphisms in the CTLA4 genes are associated with some autoimmune endocrine diseases in humans. Interestingly, some tumors and viruses may use the same pathways of immune regulation to evade immune attack. This realization has led to the development of antibodies that block CTLA4 and PD1 for tumor immunotherapy by removing the brakes on the immune response. These antibodies promote responses against tumors. Energy also affects mature B cells in peripheral tissues. It is believed that if B cells encounter self-antigen in peripheral tissues, especially in the absence of specific helper T cells, the B cells become unable to respond to subsequent antigenic stimulation, even though they are having these antigen receptors, and may be excluded from lymphoid follicles, resulting in their death. B lymphocytes also express inhibitory receptors. B lymphocytes also express inhibitory receptors that may play a role in limiting their activation and preventing responses to self-antigens. Suppression and the second mechanism of peripheral tolerance is suppression by regulatory T cells. Here we know that a population of T cells called regulatory T cells functions to prevent immune reactions against self-antigens. Let's discuss about this. Regulatory T cells develop mainly in the thymus as a result of recognition of self-antigen. As a result of recognition of self-antigens, you can see this in figure 6.21, right? But they may also be induced in peripheral lymphoid tissues. The best defined regulatory T cells are CD4 plus cells that express high levels of CD25, the alpha chain of the interleukin 2 receptor, and a transcription factor of the fork head family, called FOXP3. Both interleukin 2 and FOXP3 are required for the development and maintenance of functional CD4 plus regulatory T cells. Mutations in FOXP3 result in severe autoimmunity in humans and mice. Autoimmunity will destroy our self-antigens and self tissues, resulting in disease. Mutations in the FOXP3 result in severe autoimmunity in humans and mice. In humans, these mutations are the cause of a systemic autoimmune disease called the IPEX, IPEX, which is an acronym for immune dysregulation, polyendocrinopathy, enteropathy, X-linked. In mice, knockout of the gene encoding interleukin 2 or the interleukin 2 receptor alpha or beta chain also results in severe multi-organ autoimmunity because interleukin 2 is essential for the maintenance of regulatory T cells. Recent genome-wide association studies have revealed that polymorphisms in the CD25 gene are associated with multiple sclerosis and other autoimmune diseases, raising the possibility of a regulatory T cell defect contributing to these diseases. The mechanisms by which regulatory T cells suppress immune responses are not fully defined. The mechanisms by which the regulatory T cells suppress immune responses are not fully defined, but their inhibitory activity may be mediated in part by the secretion of immunosuppression inhibitory activity may be mediated in part by the secretion of immunosuppressive cytokines such as interleukin 10 and TGF beta, which inhibit lymphocyte activation and effector functions. Regulatory T cells also express CTLA4, which may bind to B7 molecules on ATCs and reduce their ability to activate cells via CD28. And third mechanism which is attributed for peripheral tolerance is we have second mechanism is by suppression by regulatory T cells we get the peripheral tolerance. Regulatory T cells may play a role in the acceptance of the fetus. Regulatory T cells may play a role in the acceptance of the fetus. Placental mammals face a unique challenge. Because the developing fetus expresses paternal antigens that are foreign to the mother yet have to be tolerated. There is emerging evidence that regulating T cells prevent immune reactions against fetal antigens that are inherited from the father and therefore foreign to the mother. In line with this idea, during evolution, placentation appeared simultaneously with the ability to stably express FOXP3 transcription factor Experiments in mice have shown that fetal antigens induce long-lived FOX3 plus FOX P sorry experiments in mice have shown that fetal antigens induce long-lived FOXP3 plus regulatory T cells, and depletion of these cells results in fetal loss. There is a great interest in determining the contribution of regulatory T cells in human pregnancy and possible defects in these cells as the basis for recurrent spontaneous abortion. Is there any defect in the regulatory T cells is important to check the cause of to cross check the cause of the spontaneous abortions, current spontaneous abortions. Now the another mechanism which is providing the peripheral tolerance is by deletion by apoptosis. Deletion by apoptosis. T cells that recognize self-antigens receive signals that promote their death by apoptosis. Another mechanism of peripheral tolerance is deletion by apoptosis. T cells that recognize self-antigens may receive signals that promote their death by apoptosis. Two mechanisms of deletion of mature T cells have been proposed, based mainly on the studies in mice. It is postulated that if T cells recognize self-antigens, they may express a pro-apoptotic member of the BCL family called the BIM without anti-apoptotic members of the family like BCL2 and BCLX, whose members of the family whose induction requires the full set of signals for lymphocyte activation. It is postulated that if T cells recognize self-antigens, they may express a pro-apoptotic member of the BCL family called BIM without anti-apoptotic members of the family like BCL2 and BCLX, whose induction requires the full set of signals for lymphocyte activation. Unopposed BIM triggers apoptosis by the mitochondrial pathway. A second mechanism of activation induced death of CD4 plus T cells and B cells involves the FOS ligand system. Lymphocytes as well as many other cells express the death receptor FAS. Lymphocytes as well as many other cells express the death receptor FAS, a member of the TNF receptor family. FAS ligand, that is FAS capital L, a membrane protein that is structurally homologous to cytokine TNF, is expressed mainly on activated T lymphocytes. The engagement of FOS by FOS-L induces apoptosis of activated T cells. It is postulated that if self-antigens engage antigen receptors of self-reactive T cells, FAS and FAS-L are co-expressed, leading to elimination of the cells via FOS-mediated apoptosis. Self-reactive B cells may also be deleted by self-reactive B cells may also be deleted by FAS-L or T cells engaging FAS and engaging FAS on the B cells. The importance of this mechanism in the peripheral deletion of autoreactive lymphocytes is highlighted by two mice that are natural mutants of FAS and FAS-L that are natural mutants of FAS or FAS-L. These mice develop an autoimmune disease somewhat resembling human systemic lupus erythematosus associated with generalized lymphocyte proliferation. In humans, a similar disease is caused by mutations in the FAS gene. It is called autoimmune lymphoproliferative syndrome (ALPS). Some antigens are hidden, that is sequestered from the means dead from the immune system, because the tissues in which these antigens are located do not communicate with the blood and lymph. As a result, self-antigens in these tissues fail to elicit immune responses and are essentially ignored by the immune system. This is believed to be the case for the testes, eye, and brain, all of which are called immune privileged. All of which are called immune privileged sites because it is difficult to induce immune responses to antigens introduced into these sites. If the antigens of these tissues are released, for example, as a consequence of trauma or infection, the result may be an immune response that leads to prolonged tissue inflammation and injury. This is the postulated mechanism for post-traumatic arthritis and uveitis.